Prior to the lab, agar plates were poured for the growth of the bacteria. For the S3 plasmid lab, 2 Lb agar plates and 2 Lb ampicillin agar plates were made. For the pGLO plasmid lab, 1 Lb agar plate, 2 Lb ampicillin agar plates, and 1 Lb ampicillin arabinose agar plate was made. One of the Lb agar plates was then used to streak for single colonies of E. coli and then incubated at 37 degrees Celsius overnight. In both labs, 250 microliters of transformation solution was pipetted into 2 Epidorf tubes, which were then placed on ice. About 2 colonies of the E. coli from the previously incubated plate were collected and dispersed into each tube of the transformation solution. In the S3 plasmid lab, 10 microliters of S3 plasmid was pipetted into the positive S3 tube, none in the negative S3 tube. The same process was used for the pGLO plasmid lab using the pGLO plasmid and the positive and negative pGLO tubes. After the plasmid was added, both positive and negative tubes were incubated for 10 minutes at room temperature. They were then placed in a 42 degrees Celsius water bath to heat shock the bacteria. Heat shocking causes the membrane of the bacteria to become porous, which allows the plasmid to enter the cell. After 50 seconds in the water bath, the tubes were then removed and placed on ice for 2 minutes. This allowed the membrane to become solid again. 250 microliters of Lb broth were then added to each tube and then incubated at room temperature for 10 minutes. The mixtures were then pipetted onto the agar plates. In the S3 lab, the mixture without the S3 plasmid was pipetted onto both a Lb and Lb ampicillin plate. These serve as a positive and negative control because a full bacterial lawn should grow on the Lb plate and nothing should grow on the Lb ampicillin plate. The mixture containing the S3 plasmid was pipetted onto an Lb ampicillin plate in order to test whether the plasmid caused antibiotic resistance. The S3 gene causes antibiotic resistance in the transformed bacteria, which are the only ones able to grow on the Lb ampicillin plate. Single, clearly defined colonies could be seen on the plate after incubation. The S3 gene is constitutive, meaning it does not need an effector to be active. The same process of bacterial lawn was used in the pGLO plasmid lab. The mixture without the pGLO plasmid was pipetted onto an Lb and Lb ampicillin plate. These serve as the positive and negative controls like in the S3 plasmid lab. The mixture containing the pGLO plasmid was pipetted onto the Lb ampicillin and the Lb ampicillin and arabinose plates. The presence of arabinose tests the facultative gene that causes the bacteria to glow. While the gene that causes antibiotic resistance is constitutive, meaning it is always on, the pGLO gene is facultative and requires arabinose as the effector to turn off the repressor so the gene can be active. The transformation efficiency of both plasmids was calculated, and the S3 transformation efficiency was 5,663 CFUs per microgram, and the pGLO transformation efficiency was 1,977 CFUs per microgram. An easy error to make is not flaming the inoculation loop between passes of bacteria or not using a new sterile loop each time. This does not allow the bacteria to be isolated because it drags more bacteria than needed across the plate. We did not experience this problem because we used a new sterile plastic inoculation loop each time. Another error is cross-contaminating the samples. Our results were as expected, so we did not experience this problem.